Okay. All right.
Okay, great. Okay, hello everyone. I'm very honored to introduce today's colloquium speaker, Professor Michael Snyder, from Stanford University. He's an internationally renowned expert in genetics and personalized medicine, widely recognized for his pioneering contribution to integrated systems biology and precision health. His work has significantly advanced our understanding of individualized health profile, early disease detection, and personal therapy strategies. In this colloquium, Professor Snyder will talk about disrupting healthcare using deep data and remote monitoring. So please welcome Michael Schneider.
Okay, great. Well, it's a
Uh-huh.
real pleasure to be here. I hope you guys are having a great class. Yeah, and I'm going to tell you, as the name says, about using deep data and some new technologies we've been developing and using to try and change the way we practice healthcare. So I'm going to start with the premise that many of you may agree with, which is the healthcare system as currently practiced is fairly broken, meaning, first of all, we tend to practice sick care rather than truly healthcare. And even when we practice healthcare, it's a pretty archaic way we do it, meaning what one does typically, at least in the US, is you'll typically get in a car, always at an inconvenient time to travel to a physician's office. The office itself looks pretty much the same it's looked for the last 40 years, maybe a few differences. When you're there, they'll stick a needle in you, and from all that blood they draw and the time you're there, they actually don't make very many measurements. And then from those measurements, they'll treat you based on population averages, population measurements. We think all of these methods, all these steps can be changed, and so we've been working pretty hard to do that. And just to illustrate this last point about treating people based on population averages, um, this is in Fahrenheit, but in the US, we're told that your temperature when you put a thermometer in your mouth is 98.6. But if you actually look at the data that are out there, it's more like 97.5. But the more important point is there's a spread, meaning this is the 25th quartile, that's the 75th quartile. So what that means in today's world, if this is your normal healthy baseline at 94.6, and and you go to a physician's office and they measure you at 90.6, they'll tell you you're normal, you're healthy, everything's fine. But I guarantee if you're up 4 degrees over your baseline, you're not healthy, something's off. So that's a big theme of our work. Know what your healthy baseline is so you can detect shifts from that. Now, your health is influenced by many things. It's influenced by your DNA, but that only accounts for about 16% of your lifespan. Instead, all these other factors like lifestyle, such as exercise, food, mental health, even environmental exposures, all impact your health. And we're in a world where we can quantify a lot of these. So, for example, it's pretty easy to measure exercise with activity watches, things like that. Food is still a bit clunky, but still measurable. And mental health, we now measure indirectly. And these we can measure as well. So we can quantify these, but even if we don't do it directly, we can quantify their effects by collecting deep data on people. And that's thanks to a revolution in technologies. Meaning in the last, say, 15 years or so, there's been a revolution in DNA sequencing. So you can now sequence a human genome for about $100 US dollars. The interpretation's still more on top of that. Mass spectrometry, we can now measure many thousands of molecules out of blood and urine. And the wearables we'll talk about, we do quite a bit here where you can continuously measure people with smartwatches and rings. And so, because of this, we can now collect data at a level that's never been possible. And a number of years ago, oops, this is an old slide. It's actually about 12 years ago now, we set up what we call personal omics profiling, where it will collect blood, urine, and stool from people. And then we'll sequence their genome once, but then we'll measure out their immune cells, peripheral blood monocyte cells, make as many measurements as possible. So we'll follow their DNA methylome, their transcriptome, their proteins, etc. And out of urine, we'll also measure metabolites and lipids. We do follow the microbiome, which we know is important for health. And this is on top of deep clinical tests, questionnaires, some advanced tests like stress echocardiograms. And we started with the wearables about 10 years ago, so maybe a little longer now. So the bottom line is we're collecting deep data on people, but equally importantly, we're collecting it longitudinally. So we have a cohort of about 109, started with 109 people, and we've been profiling them, as I say, for about 12 years. We collect samples every 3 months while they're healthy. And if an adverse event like a viral infection comes along, we'll take 5 to 7 more samplings. So we're doing this to try and understand what a healthy profile looks like, not a sick one. How does it change over time? What's the difference between people? What happens when people first get ill? And importantly, from the health management side, can we use advanced technologies like genome sequencing wearables to better manage people's health? And I can tell you when we started this, people did not like us, physicians in particular, did not like us sequencing the genomes of people, healthy people. They thought it was going to cost millions of dollars, turn everyone into hypochondriacs. I think they've largely warmed up to that now. But many areas they still don't like wearables and some others. And so, with regards to the health profiling, we did in fact discover just in the first 3.5 years 49 major health discoveries. So they span a lot of different areas: hematology, cardiovascular disease, metabolic, and so on. And some of these were a pretty big deal, meaning, so this, by the way, that these are all found pre-symptomatically. People did not have symptoms yet. So we caught one person, for example, with early lymphoma. These two are precancers that can convert to aggressive cancers. Two people with serious heart issues, one picked up by genome sequencing, another by wearables. And that was another lesson. No one technology found these. Sometimes it was imaging, sometimes it was the biochemical profiling. Sometimes it was the gene sequencing. So no one item found these things. It was often the combination. And so the way we like to think about it is that by doing these deep profiles, we're getting a much clearer picture of people's health than we would otherwise get. We, for example, might be seeing 500 or 600 pieces of a jigsaw puzzle versus 5 or 6, we would say, when you go to a physician's office today. So the bottom line is that these deep data profiles do reveal lots of things. Now, genome sequencing turned out to be very, very powerful. Just from the first 70 people who we looked at, 12 had something pretty important. So these are single gene mutations. And the prototype of this is a BRCA mutation. I'm sure many of you have heard of this. This predisposes people to breast cancer, women to breast and ovarian cancer, men to breast and other cancers. So we did have one individual who had a mutation here. And then we also had, though, others that turned out to be pretty important. So one person, a fairly young guy, had a mutation in this gene, which put him at high risk for dilated cardiomyopathy. And sure enough, he did a follow-up stress echo, and he actually has a heart defect. He's on drugs. Person up here actually had a mutation here, puts him at risk for certain cancers, did a whole body MRI, and sure enough, had early thyroid cancer, which was caught and then managed because we had sequenced their genome. So we wound up finding some pretty important things that were very valuable to people. Now, most diseases, you may know, they're not single Mendelian diseases, they're complex diseases. And those generally are thought to be due to many genetic changes of small effects. And so people have developed polygenic risk scores to try and make predictions. The problem is these don't work very well. So, by the way, so single gene mutations are like getting clobbered by a sledgehammer. These are more like death by 1,000 cuts. If you inherit a lot of the bad alleles, you're at high risk. Good alleles, you're at low risk. And so the net result was uh, we people do polygenic risk scores to try and predict these. The problem is they don't work very well. They only work for the top 2 to 4% of the population. Um, and we think there's several reasons for that. One is people don't, they ignore the rare variants. And the other is they actually do linear addition of the risks. And genetics is simply not linear. So we've come up with new ways. I won't have time to get into it. Sorry, here, so I'm drinking water. Anyway, we've come up with new ways of analyzing genomes for complex disease. You can feel free to look at these papers. But the net result is they work pretty well. We find with they use AI. This is a rare variant method. These are using common variants from GWAS signals. And we always find 10 to 100 times more genes than had been reported previously, and much more of the heritability. So these turn out to be very, very powerful for getting at the genetic basis of complex disease. And in one case, we've even been able to set up for AAA. This is a disease that actually kills a lot of people when their aorta bursts. So it's a terrible way to know you're at risk. In one case, with our new gene list, we could come up with a decent predictive score for AAA. So we think these could be turn out to be quite powerful.
Snyder, I'm sorry. What was AAA? Sorry.
Oh, I'm sorry. This is abdominal aortic aneurysm.
Okay. So you use only genome data to predict that risk?
We did, but if you add in a little bit of EHR data,
Uh-huh.
you can get even better. So
Ah,
you can add
okay.
multiple features. Yeah. Thank you. I didn't
Okay.
explain it very
Thank
well.
you. Thank you.
Yeah. So all these, again, I haven't gone through this, but ALS was very, very powerful. There were 7 genes found from a GWAS study. And with our new method, we found basically 700 genes and could explain 6 times more of the heritability. So anyway, they turned out to be quite powerful, these new methods. So, bottom line is what I'm encouraging you is that by applying new approaches, we can really uncover the genetic basis of many important diseases much, much better. The other thing is that because we're profiling people longitudinally, we can see how they change over time, which we interpret as aging. And it turns out everyone ages differently. So, for example, this person, their top biochemical pathway that's changing is their cardiac hypertrophic signaling pathway. They're a cardio ager. This person, their immune system and metabolic pathways change. We think you need as few as 5 data points, and we can see pretty accurately how you're changing. We call these aging patterns ageotypes. And so the bottom line is that here's a case of 4 ageotypes. Each column's a different person, each row's an ageotype. And what we found is that the people over here, they're aging in all 4 categories. This person's aging in 3 of the 4, not much of a kidney ager. This person's kidney ager, but not others, kidney and metabolic, and so on. So we can actually see how people are aging and their markers associated with these ageotypes. And in fact, in some cases, people have changed their lifestyle. I'll come back to this, and they've improved their markers. So we think this information is very, very actionable. More recently, we've asked a very simple question. Are people just aging linearly? Are there times when there are more changes than others? And what we discovered is that, in fact, there seem to be at least 2 periods where there are big bursts of changes. One in the 60s that we did expect, and we saw muscle mass and skin changes we expected, that immune changes, that was pretty key. But we also saw changes in the 40s, and that was not necessarily expected. In hindsight, I think it makes sense. And so here we also saw changes in muscle mass, various metabolism like alcohol metabolism, lipid metabolism, even caffeine metabolism. This lipid metabolism we think is quite interesting because people's fat distribution does shift as they hit their 40s. And once again, we think this information is very actionable. So there are periods where there are major, major changes occurring. One of the ways in which we're scaling this up, people say, how are you going to scale this? That's a research study. Well, we're in Silicon Valley, so what we do is we spin off companies. So we formed this company, QBio. And what they've done is they actually have basically taken a medical version of what I just showed you. And they also do a whole body MRI, magnetic resonance imaging. Now, most physicians today will tell you do not do whole body MRIs. They're worried you're going to find nodules, and it's also pretty expensive. But we think getting MRIs is very important because the issue isn't do you have nodules, the issue is do you have any growing nodules. And by taking longitudinal measurements, you can see that. And so, sure enough, what happened was by actually implementing this, we found cases of ovarian cancer, prostate, even discovered early pancreatic cancer, which is almost never found early. So, this method can be very, very powerful. then more recently, we've been trying to actually implement this concept of trying to do remote monitoring. The idea about why do you go to physician's office when you could get a lot of these measurements at home? I think you'll still go to physician's office, don't get me wrong, but you can measure yourself much, much more often with at home if it were convenient. And so we've been pushing two approaches. One is wearables, and the other is home microsampling to try to basically profile people better. Now, wearables, you may know, these are usually smartwatches and rings, but there are many other devices as well. There are thousands of them out there. And they're powerful because they measure you continuously as long as you keep them charged. And so, what we did early on when these came out as fitness trackers, we actually put them on our cohort. And we discovered right away that actually you could detect infectious disease. I forgot to say these have become very popular. So, a lot of very famous people actually wear smartwatches or rings. Anyway, we discovered quite early you could tell when people are getting ill with a simple smartwatch, in this case, a smartwatch and a pulse ox. And this is me when I figured out my Lyme disease. Basically, I discovered that my blood oxygen dropped and my heart rate went up pre-symptomatically. I was on an airplane at the time. And when I landed, it didn't come back to normal. It was abnormally low for the blood oxygen. It was abnormally high for the heart rate. It was 2 weeks after I was in a Lyme infested area and went on to diagnose it as being Lyme disease, actually. And when I got home, I got tested as Lyme positive. I've given blood before I left as negative. I was on a trip. Bottom line out of all this is that I could first tell when I was getting ill pre-symptomatically from a simple smartwatch and pulse ox. And then we went on to show we could tell when people are getting ill from respiratory viral infections because your heart rate jumps up. So we published this back in 2017, and then along came COVID, as many of you know, that hit us in spring of 2020. And these days, people try and detect COVID with temperature, which is a 300-year-old concept. And it doesn't work very well for COVID because most people don't get a fever. Or you can use antigens and PCR tests, which are more accurate, but you don't do them all the time because they're inconvenient, a little bit pricey. So we think wearables could be very powerful. So when the pandemic hit, we quickly partnered with Fitbit, enrolled 5,300 people. And the first step was to see, can we detect COVID with a simple smartwatch? And the answer is we could. We had 32 people who were wearing their Fitbit while they got COVID, and they had a diagnosis date and a symptom date. And so this is our very first case. This is resting heart rate. Here's a person, they were diagnosed with COVID. Here they got their symptoms the day before, but you can see their heart rate was up for 9.5 days prior to symptom onset. In the end, we wrote algorithms that worked 80% of the time. We can tell when you're getting ill in advance of symptoms because of simple heart rate. And then we went on to write real-time detection systems where we follow your circadian baseline, look for a jump up in signal that's above a certain number of standard deviations, and it'll set a red alert. By the way, the median is 4 days in advance of symptoms. This works really well for COVID because it has a long pre-symptomatic period. So with the real-time alerting system, that also works 80% of the time. Here's one of our first cases here, where here's an individual, their symptoms appeared here, they were diagnosed the next day, but they had a jump up a resting heart rate 3 days in advance of symptom formation. So this really does work. It works on Apple, it works on Fitbit, works on probably all devices, and even works in asymptomatic cases where people don't have symptoms. So here's one here, a Fitbit case. This person was diagnosed with COVID here, but they're getting red alerts for 2 weeks prior to symptom onset. And it turns out it's even more sensitive than antigen tests. This is me. My smartwatch turned positive before my antigen test did, actually, because it's so sensitive. Now, it's not specific for COVID. Other things can trigger these alerts. Turns out workplace stress is the number one trigger red alert. So we think these devices are very, very powerful for picking up mental health and following your mental health as well as your physical health. And so we're actually working on this harder and other infections, by the way, will trigger these red alerts. Many of these things that trigger it, you can contextualize, like running a marathon will trigger a red alert. But anyway, both mental health and physical, say viral infections will actually cause red alerts. The other thing we've been doing is trying to expand this to other areas. So, your smartwatch, if you think about, measures a lot of different things. It measures your blood oxygen. They measure conductance on your skin. They have a lot of pretty powerful sensors. So we took features in the smartwatch and used machine learning to see if we could predict clinical values. And sure enough, we could. Here's red blood cell count, hemoglobin, and others. And this is just a 2016 watch. We could easily pick up signals for these. And they're not clinical grade measurements, but they're good enough to detect your shift from baseline. So we think smartwatches are going to be powerful for measuring many, many kinds of things. The other thing we've been doing a lot with is continuous glucose monitoring. These devices, they'll measure your glucose every 5 minutes. And basically, they were being used at the time for insulin-dependent type 1 and type 2 diabetics. What we did was we put them on so-called normal people and pre-diabetics. And this is important because basically 11.9% of people are diabetic in the US and 33% are pre-diabetic. Most pre-diabetics don't know it, and they will become diabetic someday. So we think, excuse me, actually detecting these is pretty important. So we put CGMs on so-called normal people and found that most normal people were indeed normal. Uh, some of them, though, were moderate spikers and others severe spikers, just as bad as diabetic. We think it has to do with the way in which they're diabetic. And so we classified people into what we call glucotypes. We then went on to discover that different people spike to different foods, as did Aaron Siegel. This is from Aaron's work. So some people spike to potatoes, others to pasta, some to white bread, some to brown bread. We're all very different. And this is from Aaron's work. Here's a participant spikes a banana but not a cookie. Here's a person who spikes a cookie but not a banana. That's much more common. We actually discovered a lot of people spike to bread and peanut butter, others to protein bars, which have carbs in them, and most people spike to cornflakes and milk. We think that stuff's super evil. So go ahead.
Yes. So, uh, kind of how these personal differences are common? Is it very common or very rare?
Which ones? These cases?
Yeah, banana,
Very
cookies,
common.
apple. It's very common.
Very common. So
Wow,
again, a
surprising.
lot of people, yeah, they're really spikers and they don't know it. So yeah, we think it's very important to
Yeah,
know.
because typically nutrition give a average guidance for good food or bad food, but it could be very different depending on the person. So do you know why they are different?
Yeah, good question. That's what I'm going to
Yeah.
show you next.
Yeah, thank you.
We don't know the full answer, but we know a lot. And that's what I'm going to show you. Great question, though. So here's a new news study we ran recently. We had 55 people eat 7 different carbohydrates, identical amounts, but they're in different forms. So here's mixed berries, grapes, rice, and bread, and so on. And it turns out, once again, different people spike to different foods. So red is rice. Most people are like this. They'll spike to rice. Here's a person who spikes to bread. They tested everything 2 or 3 times. This person spikes to bread. That's blue. This person to grapes. This person to pasta. This person to potatoes. So again, different people are spiking to different foods. So why is this, you ask? Well, we think it has to do with the heterogeneity of diabetes. It's a very complex condition that involves many different organ systems. So it involves your liver, your pancreas, your kidneys, your brain even, and your muscle, all are involved in glucose metabolism and involves many different biochemical pathways. So insulin, something called incretin, these hormones that promote insulin and other conditions. So it's very, very complex process. So we went on and started typing people. These are in blue are normal, in yellow are pre-diabetics. Here's one diabetic, turns out. And we typed them for muscle insulin resistant, beta cell function, incretin, and hepatic insulin resistant. What we discovered is everybody's a little different, so or quite different. So this person, for example, they're very insulin resistant, moderate for beta cell defect, fine for incretin, and fine for actually, or maybe they're intermediate, it turns out, for hepatic. But you can see other people, they'll be very insulin sensitive, maybe have, well, they're normal here, but you get the idea. Everybody's different. Here's a better way to look at it, perhaps, where this person's incretin has an incretin defect. These are all muscle resistant. These are beta cell defects and so on. So again, different people with different phenotypes. We then went on to try and get at this a little better. And what we found is that for the CGM curves, everybody has a different curve. So, for example, this person rises fast, drops slowly. This person's more symmetrical, goes up slower, down slower. These are people with multiple humps, so they're a little strange. So, bottom line is everybody's a bit different. We think this might be due to the subphenotypes I was telling you about. So we took features from the curve, 14 in the end were predictive. And in the end, we could predict muscle insulin resistant and beta cell function pretty well, about zero point 0.9 actually for each. And we could even predict incretin defect in the others, not as good as these two. These two are quite good. And so now with a very simple at-home test, probably for $50, you can tell what subphenotype you are, which we think is very, very powerful. These normally cost hundreds, if not thousands of dollars, take a lot of time. Now you can do it in the convenience of your own home. So then we went on to try and see if we could explain the food a little better. And sure enough, here's insulin resistant. This is now an aggregate plot. Here's insulin sensitive, meaning this is a lot of people. And what you'll notice is that both of these insulin resistant and insulin sensitive spike to rice. That's red. But insulin resistant people spike to potatoes, not so much for insulin sensitive. And the same is true for pasta. So the insulin resistant people, again, they're more prone to spiking to some of these things versus others. More recently, we've now taken people's lifestyle information, activity, food, sleep, diet, and they're wearing a CGM and they're recording all this. And we're trying to correlate people's lifestyle, the things they do and when they do it, with their glucose spikes, and also relate it to their subphenotype. And the net result is that we could actually get very predictive, meaning um, we could predict people's, with machine learning, people's hemoglobin A1C, adipose insulin resistant, other subphenotypes with some of these measurements. And to show you some examples, if you eat your big meal first thing in the morning, you have lower glucose, whereas if you do it late at night, you'll have higher. Starchy vegetables are worse for you. Fruits are better for you. Most people don't get enough sleep, so if you increase your sleep, you'll lower your glucose. So that's powerful. So we can make all these predictions about subphenotypes. And here's an interesting one for muscle insulin resistant. It turns out that you can actually lower that or have lower values if you do more exercise, and that makes sense. So bottom line is we can start correlating lifestyle, what you eat, when you eat, what other features you do with all of these phenotypes. And now you can go on and actually use this information. Here's a company called January AI that basically uses artificial intelligence and can make predictions from CGM and actually even these days without a CGM, make predictions about glucose spiking using AI-based methods. Pretty powerful. And people lose weight if they go into this program. The other thing we've been doing a lot of lately is this home microsampling. And the idea is you can take little droplets of blood, you collect it on your shoulder these days, overnight it to the lab, and now with their omics measurements, we make about 7,000 measurements. We do proteomics, metabolomics, lipidomics, and a lot of targeted assays. So we spent about 7 years developing this, and it works pretty well. Not perfect, but there are certain things degrade, but most things it works pretty well on. So then we did some really fun experiments. We had 32 people drink this shake. It's called an Ensure Shake. I don't know if you can get it where you are, but basically, then we profile people. They were fasted at the time. We profile them before they drink the shake, 30, 60, 120, 240 minutes after. And hundreds of molecules change. That's not a surprise. And we can measure some pretty important molecules like insulin C peptide. This is one of the incretins and so on. Even inflammatory markers we can measure. And so what we did was when we did this, we discovered that everybody responds to that shake differently. So each box is a different person. This is just showing three different carbohydrates. And what you'll notice is that for people again are very different. So for this person, the carbohydrates goes ways down. For these people, it stays high. This person's kind of flat. This one goes down. This one goes down. So again, all different. We could informatically group people into five clusters, five groups. And these are classes of molecules. Gray is the average of all 32. Brown is inflammation. So the cool result here is that for this group of people, when they drink the shake, their inflammatory markers goes down. But for this group of people, when they drink the shake, their inflammatory markers goes up. Same with this group of people. So same shake, it's pro-inflammatory on some, anti-inflammatory on others. So we think this is very powerful because we can measure this. And if you think about in the US, 10% of people have irritable bowel syndrome. And now we can try and see what triggers that with this kind of an assay. The other thing we've done is this here, where we basically um, had one person who gave a sample every hour for 7 straight days while they're wearing a CGM and a smartwatch. So they're tracking all this physiology and glucose, and they were doing food logging and exercise logging. And the net result was we could actually start correlating a lot of physiology and biochemistry with their activity and their lifestyle. And so the net result was we do these timeline correlations. We can actually track people's heart rate, their glucose, their steps. These are just showing, there are literally thousands of associations. These are just showing a few hundred. Here are cytokines here. These are lipids in blue and metabolites in green. And some of these things make a lot of sense. So we'll see your insulin C peptide goes up after glucose. We expect that. But here's one that was a surprise. Alpha synuclein shows an interesting pattern. We think it's also going up in response to stress, and we can measure that. And that may be important. This molecule is important for dementia and Parkinson's. So the net result is we can track people now at a level that's never been possible. And we built a dashboard called MyPhD that pulls in your wearable omics data to be able to better display it. And so now personally, you can actually capture this information and view it and try and better manage your health. That's the idea. So this is where I see all this going. I see a world where we'll be sequencing people's genome. You can actually do it before they're born. And then together with deeper biochemical measurements and wearable, we'll be able to better track and manage people's health. And AI is going to be an important part of every single one of these steps. As I said before, the way we spin off these things, the way we try and get this out to the world is we spin off companies. So I found that 17 of them over the years. And the ones in boxes are very related to this health, to this work. And the net result is that we can, yeah, we a lot of these things can get out there. And I'm going to show you this one in a minute. This company is kind of interesting. It profiles about 650 metabolites spanning about 20 different wellness categories. Things like oxidative stress, inflammation can all show up from these deep profiles, even hormonal health and such. And so there's scientific literature behind all of these where we capture this information. We can even follow longevity. And then here's the longevity example. So, for example, this person, their biological age is older than their chronological age. And but the important thing is you can break down the age of types. You can say, all right, well, their inflammation's pretty good. Their metabolic age isn't too bad, but their heart age is a bit older. And then you use AI to take information from the patient as well as from the literature and make very specific recommendations, such as what you should eat and how to better exercise. It's not just exercise more, eat better. It really is very, very specific about what they're making recommendations. And so people actually who do take the test, they improve their health markers just after the very first test. 95% of them do. So it's really quite powerful. So bottom line, what I've been telling you is that deep data enables early disease detection. We think longitudinal monitoring is very, very important for tracking health, and it can also reveal your age type. And I mentioned about non-linear aging with birth changes in 40s and 60s. We can actually get at the genetic basis of disease better using AI. I told you about using smartwatches to detect infectious disease and continuous glucose monitoring for revealing glucose dysregulation and personalized responses to food. And lastly, I told you about the microsampling work that enables remote monitoring of biochemical activities. So I've been fortunate. I have an amazing team. They're listed here that have been contributing to this work over the years. And so they've done really a very special job. So I should have saved plenty of time for questions. It might be just as well I'm losing my voice. So I'm happy to take questions if you guys have any. So
Wow. Thank you, Michael. So, yeah, thanks so much. So, yeah, it's very inspiring talk. So, please feel free to ask a question, everyone. So you can unmute yourself and ask a question to Michael. So, by
I
the way,
hope you're all
Michael,
wearing
so yeah,
smartwatches.
maybe I can begin first. So,
Yeah.
you collected all this a lot of data from the about for 10 years. And then so kind of basically how can you make it? It's kind of I cannot believe imagine it how you successfully did it.
Well, we have people are very eager to give blood, and now it'll be even easier with the microsampling. But
Uh-huh.
it turns out it's very powerful because you really can track your health and you can see when things go off. And so
Mm,
I think that's been very, very important for people. It keeps them motivated when they see that
mm,
what changes are occurring. So it
okay.
turned out to be very, very powerful. Yeah. Now,
Nice.
had to stop during the pandemic. They wouldn't let us bring people in to collect
Right.
blood. But we started up again after the pandemic. So
I see. So so the all that data are at this point the closed, not the open yet, right?
no, no. Most people have agreed to make their data 100% open, including their
Oh,
genome sequences. And so
oh,
you can get my data. I have about 2 petabytes of data sitting there you can download and analyze. So
oh,
most of the data is yeah, it's all available. So feel free to go for it. A few people
wow.
did not agree to make their genome sequences public.
Mm-hmm.
So they're behind a firewall. You have to ask for
Wow.
permission.
Oh, I
But the
see. If
rest,
we go to the
you
My Data, we can see the data. Wow.
you can, yeah.
Amazing. Wow. Okay. So there is a question via chatting. You show the impact of genetic data on personalized health. Are you gathering proteomic or epigenomic data for personalized health care?
Yeah, absolutely. So we are doing proteomics. I didn't emphasize that. We
Mm-hmm.
started with spec. For some of our studies, we now do O-link, but we'd like to do O-link on all. We have over 2,000 samples at this point.
Mm-hmm.
So
Right.
we are collecting, and that turned out to be very, very powerful. It really does give you a much more complete picture. Now, the epigenome, we've done some
Mm-hmm.
profiling. We're waiting to do the price of sequencing has dropped a lot. So we were waiting for that because we always wanted to do whole genome bisulfite or some genome-wide scale, and that's very expensive with epigenome. You have to go deeper than a normal genome. And so we've done a limited amount. What we've done is interesting. We're eager to do the rest and then match people's lifestyle with their epigenome changes. We think that's going to be a big deal.
Wow. Okay. So any other question? Please feel free to unmute yourself.
Hello, Professor Michael Snyder. My name is Dave Kim. I'm working on the KAIST Brain and Cognitive Science Department. And actually, I have one question about your work on the wearable, the anomaly detections related to COVID-19 from the heart rate data.
Yep.
Actually, in your work, you use the resting heart rate instead of the whole time series of the heart rate. That
Yeah.
I think the
Go
reason
ahead.
why you use the resting heart rate is to remove some noise because there is a many a noise in the heart rate data. So to fully focus on the some signal related to COVID-19, you use the resting heart rate. Have you tried to use the full time series of the heart rate data to detect anomalies related to the disease instead of the resting heart rate?
Yeah, great question. So the answer is yes, we started simple for the reasons you just said. Just pick resting heart rate because it's just a lot easier to interpret and deal with. But we think that other measurements at other times is going to be powerful. You will have to correlate it with what they're doing. So, for example, we know lots of things can accelerate heart rate, like steps, for example. But that may be the
Mm,
best time to pick up COVID. You may actually see bigger changes
Mm,
when people are walking than they would if they're just resting,
mm,
if they have COVID. Meaning, so some of these things may be good stress
mm,
detect. If you stress people, you may tease out defects better. So we actually think that would be powerful to do. We're working on algorithms to do just
I
that.
see.
Yeah.
And actually,
okay.
I have a one more.